Okay, I think we'll make a start. So welcome everyone. Thank you very much for joining today's neuroscience user group meeting. I'm Melinda, so I'm the customer success manager at AD Instruments, and I was a neuroscience researcher for about 10 years doing research and teaching before I joined ADI. So I'm really excited to chair today's meeting to advance neuroscience education. And just before we move ahead, so anyone who's kind of new to AD Instruments or the LT platform, I'll just give a brief introduction so that everyone's up to speed on what we're talking about. So AD Instruments specializes in software and hardware for life science research and education. And one of our main specialties is this LT platform, so which is specifically designed for educators. So LT is actually the only cloud-based learning platform for data acquisition. So what that allows is that you can really engage students with real data. So for example, students seeing their EEG recorded in real time, and seeing how easy it is, it really enriches the student learning experience. And there's over like 900 lessons in like life sciences, nursing, medical, or veterinary sciences. But today's focus is on LT neuroscience. So in addition to the 22 lessons and tutorials, we're going to be talking about the interactive labs, lab practicals. So there are some practicals. There are 22 that are on offer in LT, and um, the focus today, or the objective today, will be to hear from these experts on how they're using, how they're engaging students with LT and their practicals they're running in undergraduate neuroscience classes. And also discuss things like challenges and pain points that comes with running these labs. And the second objective is to discuss creating new content so that we can expand the neuroscience teaching curriculum. So today we have a really expert panel of neuroscience educators. So, and first and foremost, I want to thank our panel for their time and effort for sharing their insights and ideas with everyone. Um, and first up is Associate Professor Brian Billups. So Brian is from the Eccles Institute of Neuroscience at the Australian National University. And Brian's research focus is understanding synaptic function and the role of glial cells in modulating neurotransmission. And he's also the Associate Director of Education. So he's got a wealth of experience in teaching neuroscience. And I'm really excited to hear from Brian. So I'll pass it to you, Brian.
Okay, thank you very much, Melinda. Hello, everybody. Yeah, as Melinda said, I'm from John Curtin School. I'm in the Eccles Institute of Neuroscience there at ANU. A lot of the teaching that we do in John Curtin School is mainly third year biomedical science students, right? So I'm going to show you how we use LT in our third year systems neuroscience course, and that's a course of about 80 students. So a lot of them are in their final semester of their degree, and then there's about a dozen master's students in there, so they're at quite a high level. And our aims in using LT and running the practicals that we run are really to get those students to think about scientific inquiry and how they're developing as scientists and hopefully moving on to honours projects. So this is quite challenging for them, and different people will have different examples, I'm sure, of where they're using first and second year students. But as I say, this is third year practical. So what I'm going to do then is share my screen. Let me see if this one works. Let me go that one. Okay. Melinda, can you just tell me what you see?
Yep, I can see your screen. Yep.
You can see the whole screen, and if I flick between, you can see.
That's right. Yep.
Brilliant. Yep. Okay, so what we have is we have 3 practicals that run 2 weeks apart for this group of students. And they're sort of scaffolding they get slightly harder in how we teach them. So for each practical, it's set up as a module. So this is our module. Maybe I'll go to what the students see. So they'll come in and they'll see the first one is our spinal reflexes laboratory here. And they'll come in and there's two things set up. One is a pre-practical session, which I'll show you in a moment. And then there's the actual practical lab. And you can set this up in LT so that they have to do the pre-practical before they have access to the practical lab. The pre-practical is a single user thing, so they all do this by themselves before they come into the lab. And this is a great way to get information into them, to get them thinking about the subject. We all hope they went to the lecture beforehand and actually had the material, but we know in practice that's not necessarily going to be the case. So there's the essential bits of material in there before the lab. So that's why you can see this one has a padlock on it, because they can't access this because they've not done this. The laboratory itself is a group lesson. So the students are logging in as a group, they're working together as a group, they're collecting data as a group, and then they can go away. That's all, obviously it's all on the cloud because it's LT, and then they can go away and analyze that individually. So they're working together as a group in the practical lab, and then they're analyzing and writing a report individually afterwards, and that's where they're getting their grade. So that's what they would see. So if we look at the pre-practical session, so this first one is spinal reflexes, right? So we measure EMG, we tap the tendon, and then we do that for a knee and an ankle, and then we do a Gendrasic maneuver where you go like this, clench your teeth, pull it hard, and you'll see the EMG should get bigger, but it shouldn't get faster. And there's a few things they have to learn, but it really gets them used to the LT system and links in with what we're teaching them in the classes. So the pre-practical then has some information beforehand, some basic anatomy. As it said, they must complete this work before coming to the lab, so they must complete it, otherwise they don't have access to the lab. And we also put a couple of percentage points to the assessment on there just to encourage them to do it, though it's really is formative assessment. We let them change their answers. With pretty much everybody gets 100% for that, so it's only 1% or something like that. Okay, so we can put information in there. There's questions, so if you're familiar with LT, there's typical sort of drag and drop type questions, click something questions. You can check your answer. It tells you if you're wrong. It gives you the reason why you're wrong, as I say. So it's really formative assessment type stuff. And then that's all going to be, this sort of question are all automatically marked. So that goes into the grading at the end, and we don't have to do anything with the marking there. There's information, there's notes they can make, etc. Now, the one thing in here that we have that you might want to consider is there's one question in here where we actually ask to write an answer. So we ask them to think about which spinal segments are involved in the reflex, and then we have a written answer. That's great, but when it comes to grading, you're going to have to read all these and mark them yourself. And that's actually quite a time cost, especially if you've got a big lab. So you have to think carefully about whether you want this sort of question in your assessment, because it will add a bit of time. And if you've got a class of a few hundred students, it really, whether that's worth it or not, you have to think about that. And then the other thing about having the pre-practical is we can put important information in there that we know they must read before coming to the lab. So that could be safety instructions for this lab. You know, it's in Canberra, it's in the winter, they'll all show up in their jeans and they won't be able to get the electrodes on their legs. So we say, you know, bear in mind, you're going to have to do this, come to the lab wearing shorts or loose clothing. But you can guarantee that they've read this before they come to the lab. And then they commit that pre-lab and then they get access to the lab. Okay, so when we come to the actual lab, so in there, when we come to the practical session, so this is a session that comes with LT and we've sort of modified it ourselves. So again, there's instructions to how you would do the practical. We get them all to log in as a group. I get them to say who their group members are, because when I do the marking, I sort of make sure the same markers have the same group. There are ways you can look into the system, but it's also easier if I just get them to write who they work with. So information about the setup, and then they get the data. Another nice thing, obviously, with the LT is that there is sample data in there. So if you've got students who couldn't attend, or students who were sick, or the whole thing didn't go very well and they didn't get very good data, you can put in some sample data. You can give them a code, so they only access that with a code, and then there's the data that's loaded. And you can, if you've got a Power Lab and the appropriate software, which ADI will give you access to, you can record your own sample data, and you can put your own sample data in there in the file in the setup, so it can exactly do what you wanted. And in COVID, this was an absolute godsend, because we couldn't get the students into the lab, so we could just, I took a Power Lab home, I did the prac with my kids, I got all the sample data I needed, I loaded it all up, did all the conditions, and there it is. So for a standard practical, the sample data is there, but if you wanted to do something more imaginative, you can edit that sample data, right? So it's all there, and then there's instructions how to measure it. So they do their measurements, they put the measurements into the spreadsheet. You can do some calculation. One thing I get my students to do is I give them a Google Doc. So there's a link here to the class Google Doc, so they can load up all their data into the Google Doc. So when they want to do the write-up, they've then got access to everybody's data. ADI could possibly implement something of their own. We don't necessarily have to go to an external platform for that, but a Google Doc's easy, and you just drop the URL in here, and then our LMS has the same link to the URL to the Google Doc, so I can keep my eye on that, and they can see everybody else's class data. So if they're doing a class average or some more power for the statistics, then they can look to that Google Doc. So this is one of the standard practicals, but how we run this slightly differently is we try and get the students to think about what they're doing and to come up with a hypothesis and to test a hypothesis. So this is a Gendrastic maneuver, and we pretty much give them the aim. For the first practical, we give them the aim. We say, well, the aim is to see if the effect of the Gendrastic maneuver on the reflex. So your hypothesis would be, if I do the Gendrastic maneuver, I would expect the EMG to get bigger, and we give them that. That's the first practical. We give them the hypothesis so they know what they're doing, and in the subsequent practicals, we're asking them to develop their own hypothesis and use the equipment and use the ability that we've given them to test their own hypothesis. Okay, and then the assessment is a report. So they go through they measure the conduction velocity, they measure the amplitudes, and then we say, okay, what we want as an assessment is a report, and here's an example of one. We're expecting them to write an introduction, what they did, how that led them to the hypothesis. We expect them to give an aim and a number of hypotheses, which for the first time we give them. We expect them to write a bit about the methods. Then we want them to present data as if it was like a paper. So we're asking them to pull sample raw images from the system, present it in a figure, with a figure legend, with a results section that refers to the figure, with a mean, with a standard deviation, with appropriate statistical tests, with a discussion down the bottom, with a number of references, and then we tell them to put all the data in an appendix so we can check it, or we can check the class data sheet. So this is where it's really like, third year stuff. This is the sort of capstone research experience practical that we'd expect them to be able to do this sort of stuff before they go into honours or they do their master's project to present something like this. So not everybody would want to do it like that. So within LT, then we give them the instructions, we tell them exactly what they need, and then that's just submitted as a PDF in our learning environment, which is Moodle based. Okay, so that's the first practical. The second practical goes on from this. So if we look at our second one, this is visual evoked potentials. So for this one, we have, let's have a look how we do this. Okay, so what we're doing here is we're putting electrodes on the head, we're flashing images on the screen, we're measuring the evoked potentials from the back, from the visual cortex. So obviously they've had a lecture, but we're going to present the material again so that we're sure that coming into the practical they've read it. So there's theory, there's what you're measuring, what it means, there's some formative assessment questions. Hopefully they get them all right. And then the other thing that we did here is we then, within this, we could record our own video. So again, especially this was useful for COVID. So as they're coming into the lab, I mean, it's great if you've got great demonstrators showing them how to do stuff, but if they've seen the video beforehand, then, you know, we filmed the video of the setup, and when it comes up, sorry, I'm at home, my internet's a little slow, but this is me in the practical lab having somebody stick the electrodes on the head, and we're showing them, you know, in the right way. So this would be the sort of demonstration we would do at the start of the lab anyway to the whole class, but they've already seen the video and they've had a few of the tricks, and, you know, we've done that, we've edited it. Obviously we only had to do that once a few years ago, and it's up there, and we just have the link in LT to the video, so that's great. And again, in there, we've got the vital instructions. You know, we tell them beforehand that we're going to put gel in the hair, so make sure that, you know, you're prepared that you're going to get messed up in the lab and we're going to put gel in your hair. We don't want people showing up to the lab and being surprised about what they have to do, especially the next lab where we've got live cockroaches in the lab. We don't want too many people being shocked by what is actually going to happen in the lab. Okay, and then the lab itself is a similar format to the previous one. Log in as a group, collect data as a group. You can analyze it on your own afterwards. So instructions on how to set up, instructions on how to put the electrodes, sample data in there if they need it. But this time the difference is we don't tell them the aim and we don't tell them their hypothesis. So we've given them the tools to investigate these potentials, and we've got the demonstrators to give them ideas, and they can do various things. An obvious one is if you look at the visual effects binocular, both eyes, or you cover one eye and you look monocular. And there's a difference in the speed of processing between the monocular and the binocular, so they can do this, they can do the measurements, they can do a t-test or other statistical tests. They can look at the class data, they can look at males and females, there's differences, males or females. They can take somebody who's got glasses and take the glasses on or off, but they're coming up with the hypothesis themselves. And then in the report, we're expecting the introduction to explain that hypothesis. So if their hypothesis was males and females are different, then the introduction is going to have some references as to why, what's been studied about this before, what do we know. They can't just come up with the hypothesis out of thin air. The introduction has to lead to the hypothesis. So that's how we're sort of developing their skills and making them think as researchers. But conceptually, beyond that, it's roughly the same. It's the same sort of thing. Again, we have a report at the end. When we do the reports, we have demonstrators mark the reports. They give feedback. I make sure that the same person is marking the 3 reports. The feedback is sort of scaffolded, so they're not really used to this sort of report. If they're making mistakes, we cut them some slack in the first one. The marker will say, yeah, this is good, but, you know, your figure legend needs to have a title at the beginning, or you need to say whether this was a paired or an unpaired test, whatever. And then the same demonstrator, same marker is going to get that student afterwards. And if they've ignored the feedback, then they're going to start losing marks for not saying what their N number was, or whether it was an SCM or an SD, or how they've laid out their results. So that's quite important, we find, for developing how they're writing over the 3 practicals. And then finally, just to finish off then for the third one, this gets even more challenging for them, and this one is cockroach sensory nerves. So for this, we buy cockroaches from the local pet store. We anesthetize them, which involves putting them in the freezer. We then stick electrodes in the cockroach leg. So it used to be, when we had more time, and also if we could get really big cockroaches, the students would dissect the cockroaches themselves. That was when we had a researcher in ANU working with cockroaches, and he could grow us nice big ones and supply them. Now we get them from the pet store. Sometimes they're a bit small and it's a bit fiddly, so we tend to get the demonstrators to do the dissecting the cockroach leg. If you can get nice big cockroaches, it's okay, but otherwise it can be a little challenging, because you've got the leg here, and then you've got to get 3 electrodes in the leg and not short the electrodes out, and it can be a little bit fiddly to set up, but that's real life for students, and we want them to get used to the idea that they're recording action potentials. Sometimes it works, sometimes it doesn't work. Sometimes they're bigger, sometimes they're smaller, sometimes there's electrical issues, sometimes there's noise, they have to wiggle cables. If it doesn't work, they have to give up, get another cockroach leg. You know, they have to know when to make that call. So in the pre-practical, they're told what they have to do. In this one, we're talking about bar graphs and histograms as ways of presenting data, so I introduce that to them. Again, questions. They've got to understand that they're recording electrical extracellular action potentials. That's not the same as an intracellular action potential. They've got to understand where that signal is coming from. They should understand that every action potential is the same size, and if they've got big ones and small ones, they're coming from different nerves. We're separating those out with histograms. So there's several complex concepts that they really have to get with this. It's really challenging for them. And then again, when it comes to the actual lab, first they've got the challenge of the difficult biological prep, the challenge of the conceptual stuff, and also then the challenge that they have to think of their own hypothesis. And we lead them through a few different things they can do. They take different spines and wiggle them. We look at sensory adaptation. So we bend a spine. Does it fire action potentials more at the start of the stimulus versus the end? So they're going to have to measure the frequency at the start, the frequency at the end, see how that changes over time. And that links in with the sensory adaptation lectures that they will have just had the week before. So yeah, it's challenging, but this is, you know, coming towards the end of their last course in the 3rd year. And again, they're writing up a report. They can choose their hypothesis. The hypothesis might be easy, you know, is one spine different to another spine, or the hypothesis might be difficult about sensory adaptation. And in a way, they can choose their own adventure. They can make it as hard as they want to. The class data spreadsheet isn't very useful for this because everybody's cockroach leg is different. So I don't even give them a class data spreadsheet for this. Yeah, and that's how it goes. So they do those 3 practicals. After the first practical, I give them a lecture about what I expect on the write-up. So I run through all the analysis, I run through the write-up, and I help them as much as possible in that. And then after that, it really comes down to the markers' feedback on the reports and any questions that the students have. They obviously then they email the demonstrators and they email the lecturers if they have questions on the material or on the practical. But overall, the feedback is excellent. They really like getting into the lab. They like doing the practicals. And the students enjoy this part of the course. So, yeah. Any questions from the other panel, or anything that, Melinda, you've got from the chat?
Yeah, no, there's no open questions. Yeah, feel free to ask questions. I think Brett, wants to jump in.
Fine. Yeah. Thank you very much. I mean, as you'll see when we present where we're at, it's incredibly powerful to see how you're using the system. So just some tangible questions first to get our head around the logistics for you. So you have demonstrators in the room when the students are running through the labs.
Yep.
Do you have how big is the total group in the room at one time? How many workstations do you run? Do you do
So
repeats?
we have 20-something workstations.
Okay,
And that's just because we have that many Power Labs.
right.
They're all connected wired at the moment to the internet because they're all pushing data up to the cloud. So if you're going to run that with a Wi-Fi, it's got to be a good Wi-Fi. Because we're digitizing traces, so obviously that's pushing data up at quite a rate. And then our class size varies, but our maximum is 80. So then you've got 4 people to a group. Sometimes it's 2 to a group. We let them self-select their groups. If our class size goes much higher, and it will in coming years because of changes in our program, then we're going to have to run it twice, I think, because we don't have enough Power Labs and computers, and we don't really. A group of 4 isn't ideal. A group of 2 or 3 is better. But everybody's writing up individually. They're collecting the data together, and there's enough time for them to swap over and each have a go at being the one with the electrodes on. We run the labs for 4 hours. If they can get out in 2 if they're efficient and know what they're doing. Some of our other courses are running a 4-hour lab, and then the first group in the first 2 hours and the second group in the next 2 hours. So it depends on how complicated you want it to be. But also the students often like to stay behind and do analysis in the room. Even though they could do analysis at home, they are actually doing analysis in the room, and the demonstrators are still there. So they're still helping them and get around the concepts. Now, if we were time limited, we could push them out of the room for the other group at that point. But, we like them to stick around and to chat and to discuss with the demonstrators, because they don't get many interactive experiences in their courses. So the more we can do that, as long as we can afford to pay the demonstrators, then we're happy to do that.
Yeah, that's a good point, paying the demonstrators. Sorry, did I miss your student to demonstrator ratio, or?
We tried to do 15 to 1. So
okay,
that's our sort of standard, 15, 16, maybe 20, depends on how we do it. But also we have a turnover with demonstrators, and we like the demonstrators to know what they're doing. So often we have experienced demonstrators in the room. We tend to use PhD students, but I know if some are coming to the end of the program, they're not going to be there next year. I'll get new people in who don't really know what they're doing, but I know they'll be the demonstrators next year. In previous times when we had a bit more capacity, the people who were going to do it next year, we actually put them in as students and said, right, you do it this year. If you can do that, that's great. But yeah, that's a capacity and an issue, and obviously you're paying them their time. So if you can do that, that's wonderful.
Yeah. And was there an academic in the room as well, or is it run? Yeah, there is.
Yeah,
sorry.
so there's an academic in the room, and and some of our demonstrators are pretty experienced as well.
Yeah, cool.
Do you mind if I drop in with a question about your visual evoked potential lab?
Sure.
Excellent. So yeah, we've started trying to add this in. We have to do it as a demo at the moment, just due to the amount of light pods we have. But how did you go with making the electrode placement, I guess, as robust as possible? Because I really like the video guys you had, but we had a lot of issues with trying to let the students do the electrode placement their own, especially with the electrode on the back of the head. Did you have to do any troubleshooting around that?
No. So the issue is, if you know about visual evoked potential, so we've got one electrode on the front, we've got an earth electrode somewhere. The electrode on the back of the head, what we say is you find the inion and then you go down, I think it's down a couple of centimeters, and that's where you put it. The problem with that is that electrode moves up and down. The signal will change from being an outside one to an inside one. So there is a sweet spot where you pretty much don't get anything. And it's just a bit of troubleshooting, basically, of
you just have to give
around trying to get a good connection with it. So you really need to clean it and get it on the scalp, and then it's just sometimes it works better than other times. And the system is set up so that it averages 100 flashes. And some of them are great and some of them are not so great. But we always have that sample data in there. And if a student, if they collect a lot of data, then they can't access the sample data. So then I run another lesson, which is called the sample data lesson. I give them access to that. They can go in, they can get the sample data, and then they've got something to measure. And this was when in COVID, I did my own sample data, and I think I did monocular and binocular sample data. So they've got that, and I know it gives the right result because I made sure it gave the right result.
Maybe, Brian, could I just ask also, so you've got 3 labs that run through semester, and then you've got a lecture content. Am I right in it, taking from what you said that this is an in-person lectures, so you're lecturing in a theatre?
We are doing in-person lectures in a theatre with a recording. Depending on when the lectures are, attendance might be 10%, might be 30%, but it's never 100%, right? So that's why we really worry about, giving essential content, and also timing. The lecture for the VEP, the pracs on a Monday, that lecture's on a Friday beforehand. They might not have gone to the lecture, and they might not have viewed the lecture. So yeah, it starts to get a timing. Because we team teach, everybody's a researcher, they're away, they're at a conference, then the labs are at a particular time, and we know a year in advance when the labs are, but lecturer availability is a little bit difficult sometimes, so things get switched around. So we don't want to rely too much on the attendance at the lecture for informing the practical.
Right. Thank you.
Any other questions from the panel? I had a question, Brian. Like you mentioned, you gate the lab with a pre-lab.
Yeah.
Do you get any complaints from the students, like, "Oh, you're making us do homework, like
No.
before coming in, like,
No, it doesn't.
we want to do it after"?
Sorry. And I don't get any complaints with it, and it's actually really useful in that it forces them, because if they're not on LT for other courses, it forces them to get their account sorted and get the logon sorted.
Oh, okay.
Because we found if you didn't do that, then they show up to the lab and they've never logged on, and they're spending 5-10 minutes at the start of the lab clicking that email and getting the invitation and sorting the logon. But now, firstly, we have single sign-on, so they can log on with their ANU credentials, which is great. And also I can see who has done the pre-lab and who hasn't done the pre-lab. And the ones that haven't done the pre-lab, I can send them an email and say, hey, you've not done the pre-lab. You won't be able to do the lab unless you do. Got to make sure you're on there and you've done it and you've logged in. Let me know if you've got any problems. And then that catches them all before it gives that extra little bit of engagement for them. And, and they might write back and say, oh, I'm going to drop the course, but at least you've had contact with them and And yeah, so it works really usefully, especially before the first lab, to have them all logged in and all got their accounts sorted.
That's cool. Nice. Yeah. That's a really nice fail-safe. I didn't think of it like that.
Yeah.
Yeah. Cool. So I guess let's move on to the next speakers. So we have Professors Alan Brichter and Brett Graham from the School of Biomedical Sciences and Pharmacy at the University of Newcastle. So Alan's research interests are in the anatomy and physiology of the peripheral and central vestibular system. And Brett is an expert in spinal circuits. And together they're developing a brand new third-year neuroscience course at the University of Newcastle. So I really want to hear their plans and how they're gonna crack this. Over to you, Brett and Alan.
Fabulous. Thanks, Melinda, and thanks for this opportunity. It's really great to have this group together because, as Melinda's alluded to, we are really developing something new. And I even thank you, Brian. Looking at what you've done, I feel like our ambition is way, way too high. But nevertheless, we'll see how we go. So I'll take the lead for a little bit of the presentation and give you a little bit of a background on where we're going and what our experience is. And then Alan's certainly been a key person who'll be teaching one of the modules in the lab, and we've workshopped that module, and so he'll tell you a little bit about his thinking there and how we're going to use it. So I'll try and do the famous share. I've got some slides for you, so maybe I should do the share first, actually. Am I allowed to share? Hopefully I am. Okay.
You should be, yeah.
I think that's the right one. Okay,
yeah, we can see. Yeah,
everyone can see that. And then if I can get to the little bit that makes the presentation mode. Hey, is that good for everyone? Do you see the full slide, or do you see the notes version on?
Yeah, we see the notes version.
Okay, I'll switch there are no notes as it so happens, but I'll switch it so that you don't need to see that. Is that better?
That's, yeah, cool. Looks
fabulous. Good. Okay, so what I guess a bit of a background and something Brian mentioned really is the key motivation for us to move to using the LT platform, and that is diminishing attendance at lectures. So our neuroscience lectures, we almost got to a point this year where we had single or one or two students in the lecture theatre, and it's of course a little bit demoralizing, and a lecture in that sort of format, it isn't educationally really well set up to produce an online module, which is how the students were treating the course. And so what we decided to do is to flip and to move to the only face-to-face teaching being in the lab. And our motivation for that is that we run a second-year integrative physiology course, which has that format, which has been really well attended, and really the feedback's been excellent from the students. And so we want to move our neuroscience teaching to this same format, which is basically this prac-based LT online learning platform format. So just a little bit to give you the context about the course that we do teach using the LT system and have done since pre-COVID. I think maybe 2018, maybe it was 17. And that's a second-year course called Integrative Physiology and Biomedical Science. It's taught into our Bachelor of Biomedical Sciences degree in second semester, second year. But it's also taught or can be taken by our medical engineering students, so MEdEng, in the same year. So I believe it's also in the second year of their enrollment. On average, we have about 100 students as our cohort, and the lab face-to-face opportunity or teaching is supported by online video lectures. And so again, much like what Brian said, the hope and desire is for those students to have access those lectures, which are content lectures, before they come into the lab. And there's 2 hours usually, so 2 recordings. The face-to-face prac lab happens each week, and they're 3-hour labs, so they're not 4 hours. But we do repeat. So we have smaller groups, so we only have 24 students in the room at any one time, working across 6 to 8 lab stations, and we don't really enforce group size. I mean, usually we recommend, depending on the lab, they might need 3 because they'll need a participant, as well as someone driving the equipment, and then someone setting up transducers and things like that. So on average, they end up sort of assembling into groups of 3 to 4 usually, but we don't heavily enforce that. And just by way of, you know, the mantra that assessment drives learning, just a little smidgen on what assessment we do. So we have that same pre-lab format that Brian introduced to us for his neuroscience pracs. There's one every week, and so again, this is a 12-week course with 12 labs. They do the pre-labs, and every second pre-lab that they do, they get 10%. Okay, and that's just answering the multiple choice or true false questions about the lab, as well as about the safety and the operations of the lab. And so in that way, what we've done is we've covered off any sort of safety or risk assessment in advance, and we really sell that to the students, that you must do these because you need to have looked at those documents and considered that before you enter and start in the lab. So they get a free 10% there. Our assessments are the same in that 3 of the labs are accessible labs where the students work in the lab as a group, collecting data, and then they log on individually at home. However, the lab report is quite different to what Brian described, in that really the lab report is just going through and doing the analysis of the data as well as answering those questions that are interleaved within the lab. So they do that individually and then they commit it as an individual, and then that's marked by the course coordinator actually, who marks them all. And as Brian said, the risk is if there's too much free text options in there, then that can be laborious. Usually we only have 1 or 2 free text questions and limit the characters that they have to use them. So that gives them another 30%. And then the final assessments, we assess halfway through the course. They have their first mid-semester exam, which is multiple choice. This is a face-to-face in an exam room. And then they have short answer questions, and they have 5 options. So the short answer questions are about one of the labs they've done. And they have to pick 3 of the 5 and they write about them. And so they are hand marked as well. And that covers off the first part of the course. And then we assess the second part of the course in week 13. So we assess weeks 7 to 12, and it's exactly the same format. So we've come out of this thinking that it's a really nice format for teaching. We have 2 academics in the room. And as I say, there are 4 repeats. So the labs run 4 times. So for me, that blocks out Thursday and Friday in semester 2, where morning and afternoon we're teaching these groups. And it's pretty much self-led, as you would have seen from Brian. So the students have all the instructions in front of them, and we're really moving around the room, helping them troubleshoot, laughing at the fact that they're not rescaling their axes and they can't find the data when really it's just off the screen, which still happens in week 12, which is amazing. But it's enjoyable to teach them that. I mean, it's a real important learning experience in how to collect data. So yeah, look, we think it's a fun way of teaching. That's enough about not neuroscience. I appreciate we're here to talk about neuroscience. So actually, maybe I'll really quickly flick through this in case there is any lab here that someone's interested in implementing. These are the labs where we have experience. So this is the integrative physiology course. We start the semester with what is physiology, and really we're just using the finger pulse transducer, so they record their heart rate and look at heart rate variability. Then we go into cardiovascular, where we use the finger pulse, and we also measure blood pressure with a cardiac mic on the over their radial artery, so we can digitize that. Second week of cardiovascular, we do 3-lead ECG, as well as using the cardiac mic, this time over the heart, and the finger pulse. So interestingly, we're looking at heart function across 3 different parameters that we're measuring. Then we go into a two-week block on respiratory. Week 1 is the respiratory belt. So we're looking at breathing rate, and then we also record finger pulse. And so we see some nice respiratory coupling with heart rate, which is really cool for them. The second week, we use the spirometers. So we do spirometry, lung volumes, flows, things like that. Then the next 2 weeks, and this is the challenge with physiology, there's not an easy thing for them to do in renal or gastrointestinal. Back when most of us were undergrads, we probably might have done something where we might have drunk a lot of water and then needed to collect a urine sample. That's just logistically not so easy today. So for those 2 weeks, they actually work in the microscopy labs and they look at slides of those tissues, take images, find different types of cells, and that ties to the lecture content. So there, again, that's LT based though. So Phil, who's the course coordinator, has embedded everything within LT. So they're still using the microscopy lab, taking a photo, then uploading it into LT and annotating where cells are and comparing and cross-referencing with some material that's also in there. Then the back half of the course, we do 2 weeks of metabolism and exercise. So the first one, they're all exercising on an ergometer, a cycle ergometer, doing spirometry, measuring respiratory function, heart rate, and doing blood pressure measurements manually. And then we do a VO2 max the following week, where we just have a single participant that does a VO2 max test, which is a fabulous way to teach things. Most relevant for this meeting is that in weeks 10 and 11, we do some neurophysiology. So in the first week, we do the EEG prac, simple electrode setup that we saw with Brian. Look at alpha beta, eyes closed, open, play some music. So we play like some classical music and then some Slipknot, some heavy metal music. And we look at how the EEG responds to that. And then we do things like arithmetic challenges to try and enhance cognition. And then in the second week, we use this the reflex prac, and we do the patellar tendon stretch reflex that Brian described. We also then do a ball catch with EMG on the biceps and triceps. And then we muck around with the weight of the ball. And we do it blindfolded. So at some point, we'd really like to develop a set of tennis balls that are different weights to be able to trick get into that prediction and what it actually takes to catch a ball. So that's a neat lab that the students love. And then finally, we finish with the dive reflex because we're doing comparative physiology. So they put their faces in a bucket of cold water and we record cardiovascular parameters, as well as a surrogate measure of peripheral vasoconstriction. We use the respiratory belt to measure calf diameter. And then we basically use a large leg sphygmomanometer pressure cuff to cut off the circulation. Then we release it and we watch what happens. It's really neat. Okay, but to what we really want feedback on and what we're planning. So as I said, I feel like it's ambitious, but we want to replicate that model. So we want the only face-to-face being every week in the lab on the LT platform. However, we have some constraints, and of course, we have a course design that we're looking to retain. So the first 2 weeks, for example, week 1 is in the gross anatomy lab, where they go all around and get a quick tour of the nervous system and those tissues on cadaveric tissue. And that's been in place for a long time, and we're not going to deviate from that. We're keen to implement maybe using the LT system because we have iPads in the lab. That can be used by groups. So they could walk around the lab with the iPad, and we could use the brain structure and pathways lesson within LT as kind of a guide, and we might be able to marry that up with the exercises, because again, in the gross anatomy lab, they have sort of workstations where they might have brain, spinal cords, other material that's set up, and the groups move between those stations. So we could have those stations aligned with some online content that would be on their iPad at that point in time. Sounds like a big job, actually, so who knows if we'll actually be able to implement that in time. Week 2 is then moving into the cellular anatomy. So they're in that microscopy lab, which Alan runs, actually. And so we note that there is a neural histology learning module within LT. And so one option that we're considering, again, maybe naively, is how we might marry that with what Alan does. Al, you might like to say a few words about what they do in that.
Yeah, sure. Thanks, Brett. And thanks to everybody for attending this session. I think it's going to be great to sort of establish friendships here that we can use as sort of a brains trust for those of us who are just starting out. Brett has used the LT platform for a number of years now. I feel like a Johnny come lately. I'm only just beginning to see the power of the LT system. But insofar as sort of migrating, that's exactly what I'm going to have to do. Well, not have to, but want to do with the histology. Right now, we have a reasonable system. We've digitized all our histology slides. And we have it up in the cloud through something called Sectra, where they tend to deal with anatomical specimens and histology, which means the students can access these digitized slides, high-resolution slides, and they can sort of have labels on it or no labels on it, depending on their understanding and knowledge. But what I essentially do is really act like the pilot, taking them through the tissue, highlighting the important points. But it's very didactic. It's very sort of almost lecture driven. And I think the LT system will allow us to insert more sort of written and visual material than we can simply by just showing digitized slides with annotation or not, and having a background voice, mine, actually explaining the tissue. I think the additional material that we can incorporate will be incredibly valuable. So that's the idea. As Brett said, it's sort of how shall we put it, the goal. Whether we get there this year or next year, it depends on how much time we have as our workloads increase astronomically. You're wondering how much time you have for development these days. I'm sure everybody's suffering the same issues at their institution, but that's sort of where we're heading for the histology lab, which is part of that neuroscience course.
Fabulous. Thanks, Al. So now the rest is of the course is really, we've identified labs that align thematically with the content and the organization of the course as it sits. And I should say the background of the course is that we have some neuroscientists here at Newcastle, and teaching is sort of tailored in some way to their expertise and their research. So in some ways it's an atypical neuroscience course, but I think generally it's organized the way most would be. So we move from microscopy into some electrophysiology, neurotransmission and synaptic transmission in week 3. And so I've piloted and used the earthworm action potential prac a number of times over the last 3 months or so, and that seems reasonably robust. And so our proposal is to do that again, invertebrate, so that steers us away from any sort of ethics that we need to tick off. We still have some safety that we've got to cover off, but we're going to use the existing lesson. I don't know if we use the whole lesson. We only have 2 hours timetabled for these, so we may need to, again, keep our ambition somewhat constrained, but certainly that'll be the platform. Melinda kindly came up to Newcastle and we talked about it. And one of the nice things, if anyone's run this lab previously, that he's helping us, or he has helped us implement, is averaging of the sweeps, which wasn't part of it before. And so you can imagine when you're getting a stimulus evoked extracellular action potential that's sort of jittering around, actually the learning for the students in what the raw data and the average data looks like and what signal averaging does for you is really quite powerful, I think. So we're really excited about that opportunity. We're also thinking, and again, in 2 hours, we'd probably only be able to record an action potential with the earthworm if we then move to doing the nerve function lab, where we actually put the electrical stimulator on a participant and record, again, the evoked action potential in a human. I think there's a nice alignment there to have done a cellular-based prac and then something on themselves. I'd love 2 weeks so I could do each of those pracs. I don't know that Doug, who's the course coordinator, can give me 2 weeks because he's trying to cover the rest of the material. So we'll do that one. And then our proposal, actually, it's great to hear, Brian, that you run the cockroach sensory prac. That's the one we're proposing. I'm nervous about it. We've got crickets on tap at the pet store. I couldn't find a cockroach supplier locally, and I'm still yet to try the prac with crickets. So that will live or die by the success of our trials. I could step back from that and move the nerve function in participants to that 2nd week, because really we're talking about electrical signaling in the nervous system for those 2 weeks. An adjunct to that, and Melinda mentioned that another interest is developing new pracs. So one demonstration when we used to have the lecture-based content that I used when I was teaching the students was this sensory illusion prac using hot chilies or capsaicin. And so what I would do in front of the class is kind of put a thermometer in my mouth and show them what the temperature is. Then I drink a hot cup of coffee and show them that I feel hot in the mouth. And here we go, here's the temperature. There's alignment between experience and the actual state of the mouth. Ice cubes go the other way. And then I'd eat a hot chili in front of them, which they all liked watching. I would have liked to do it. I was worried about ethics implications and whether or not I'd be allowed to just get a participant to eat a hot chili. Maybe I would, I don't know. But the point is, I would then report to them that my mouth was burning and show them that there was only really a little blip in the temperature in the mouth. And that allowed me to talk about the fact that capsaicin is activating the nociceptive pathway, the heat pain pathway, which was driving my experience, even though my mouth wasn't temperature that was driving that experience. And so that was really cool, and it aligned really well with my pain and somatosensation lectures. I wonder whether or not I could do that in the lab. And each group could actually measure their mouth temperature and then move through those paradigms, and then whether or not I'd be allowed to, in a voluntary manner, get a student that was prepared to eat a chili and then report it. I think that would be a nice novel sensory lab, and I don't know. I have looked through the LT platform. I don't think there's a lab in there that's identical to that. Maybe putting the thermometer in the mouth, sterilization, they might be some barriers. Anyway, in terms of our ambition, I don't know that's really feasible for next year, but I could do it in front of the class again. That would probably be my fallback for next year. Okay, let's move along, because I now get to throw back to Alan. So we've trialed the electrooculography lab, which aligns really well with his vestibular lectures, and so that seems to work well. But Alan's got some modifications that he's worked with Melinda on that I think we're going to be able to implement. So Al.
Thanks, Brett. And again, sort of a nice sort of gentle introduction to LT, and also the power that this platform has. And what was really good for me as a newbie to the whole system, as many of you might be, if you're using LT for the first time, is its ease of use. I think Brian obviously alluded to that too. And with Brett's integrative physiology course, it's great that some, or maybe even most of the students, actually have prior knowledge of the platform, so they're comfortable with it. But even if they weren't, even if you're introducing it to your students for the first time, I think you'll be pleasantly surprised how easy it is for the students to guide themselves, mostly through the various modules or pages that make up the module. In this case, what was great to see was the EOG lab, which is really the electrooculography. And you might think, well, what does that have to do with balance at all? But if I tell you that there's an intimate relationship between our balance and our eye movements, then you'll begin to understand why I got very excited about the EOG labs. It's a very easy setup. You just need 3 electrodes, and I forget what the module is that the
pod, which is actually the image there. Yeah, so that's the EOG pod.
So the pod was obviously essential. It is an accessory that you may have to buy extra if you don't have it already. But setting up the electrodes were even easier than what Brian demonstrated for the VEPs, or the visually evoked potentials, because they're all at the front here. Really what they're doing is sort of capturing the muscles, the extraocular muscle movements, and so it really does give you quite a nice and demonstratable, or I would say foolproof, fail-safe, whatever way you want to put it, option of detecting eye movements, which is just fantastic to demonstrate that close association with the vestibular system. So initially, the EOG lab is great. It just shows you what it's picking up, how it might be influenced by people maybe clenching their teeth or closing their eyes very tightly. That's fine. That gives the sort of nice background to what they're recording from. But then what we want to do, or what we're in the process of doing, is modifying that EOG lab to incorporate specifically vestibular or balance related activities, and it's actually quite simple. All you need is a chair that swivels, right? And it doesn't have to be too much either. What you can do is really ask the subject to close their eyes and maybe predict what do you think their eyes will do if someone's just moving them from side to side? And I'm really demonstrating how fast it happens, that you don't need anything faster than this, right? And the right answer is, of course, you have something known as the vestibular ocular reflex, which works whether you've got your eyes open or eyes shut. So it's especially good when the eyes are shut because there's no visual override. You can override the vestibular ocular reflex, but because your eyes are shut, you've got nothing to focus on. What your eyes do is move beautifully in the opposite direction of the chair movement. So if you move the chair in one direction with the eyes shut, the eyes actually move in the opposite direction, a bit like a doll's eyes, you know, the old-fashioned dolls that used to move in the opposite direction to where the head moved. So that was that. Again, I think Melinda was there, and I think the other two folks from ADI were there, and it was very easily demonstratable. And you can leave it at that, but what we went on to do was something a little more experimental, perhaps, and a little more dramatic, I think, which is that what we would try and do, and this requires a few hands holding the cables, but what we would do, again, asking the person to close their eyes, is actually rotate them in the same direction 3 or 4 rotations. Now, it's true that the wires do kind of loop around a little bit, but that they are long enough to allow you simply to rotate the subject evenly and fairly slowly around, less than, you know, sort of taking, I don't know, 2 or 3 seconds for a revolution. It doesn't have to be that fast. What would you'd pick up during that experiment is that the eyes are moving in the opposite direction, but when they reach a certain distance, they have to flick back and start again the slow process, that sort of nystagmus. And you can pick up this sort of sawtooth arrangement on your electrooculography trace, which is nice. But that sawtooth has a certain directionality to it because there's the long phase and then the short phase, right? Remembering that the sawtooth is going in one direction, fine. What happens is you'll see it over time after the second or even the third rotation that sawtooth diminishes until it becomes 0. It's because what's happening is the fluid in the inner ears is catching up with the head. And now what's happening is, and quite often this happens to pilots once they're in a loop, is that they lose the sense that they're actually going around anymore because the fluid is caught up in the balance organs. And so there's no differential between the fluid and the organs. So according to the balance organs, you're not moving your head in space, and yet you are. You're still rotating. And then the exciting part is when you stop the subject, right? The fluid keeps going. So suddenly the sawtooth actually changes direction. It reappears and it goes in the other direction because what the the client or subjects, that's the right word, suddenly feels is that they're being moved in the opposite direction, and yet they've been stopped, right? So sorry for the elaborate description, and I'm not showing you anything at the moment. It's because we haven't recorded it. The point being that this electrooculography lab has an option to become more sophisticated than it currently is. It's great as it is, that's fine, but because I happen to have an interest in balance-related issues, it's a perfect demonstration for the whole class on how this close association between balance and eye movements is interlinked. It used to be I had to demonstrate with sort of goggles, and we had infrared cameras in the goggles, and so you could actually see the eyes. And I'll still retain that as a teaching aid, a bit like Brian showed a preliminary video. But what I would like now to do is for the whole class to experience on someone they know that they're sort of actually stimulating, that it happens, and it's a reflex that we don't think about because most of the time it works so impeccably well. So sorry about sort of extending this session to be purely didactic, but the point being is that we've got the opportunity to expand on labs that are canned already, but could provide even more richness along the way. Over to you, Brett.
Thanks, Al. Now, Melinda, you might even want to jump in now and stop us. We're both very enthusiastic about the opportunities, but we don't have detailed descriptions. So maybe if you just give me a minute or two, I can just introduce the other labs we're proposing. You'll have picked up on some of them already. So you can see that there's a canned auditory lab with tuning forks that we're proposing to use that'll align really nicely with some auditory lectures. The red is where we have to do stuff. You can see there's lots of red on the second page. In motor, we're proposing to do the autonomic nervous system module, and that's because in second year in that integrative physiology, they've done a lot of EMG. So they've already done a lot of skeletal muscle stuff. So we like that. Then we're sort of going off LT, although we'd like to use the platform. So there's a week that talks about dementia, memory, and things like that, where the lecturer, who's actually the course coordinator, Doug, is proposing to introduce some chat GTP content, which we've all probably been pushed to introduce that AI module. So he's proposing that students or groups ask ChatGPT to come up with a cure for Alzheimer's disease, and then get that out, and then critique it and discuss the outcome. So that's the overall concept for that. Week 9, we have a lot of stroke researchers here, and there's a week on stroke, which we will scaffold the LT support for that around the blood-brain or the brain-blood supply lesson that's already in there. However, being stroke researchers and clinicians, they're going to embellish that with some more information and scans. Week 10 is sleep and arousal. Melinda has kindly worked with us, and we're proposing to use the LT reaction times lab. So again, if you're sleep deprived, that's going to impact your reaction times. And Doug, who's also teaching that, the course coordinator, is going to get the students to keep a sleep diary for the prior week. And so we might catch some people who are a little bit sleep deprived, and we can look at their reaction times. We'd also love to put some more EEG in there. And so we're talking about doing a 4-lead EEG, and actually getting the LT system to monitor theta delta with a person that's relaxed, eyes shut for maybe 15 minutes to see if we can get them to a state where those rhythms start to emerge. Week 11 is psychiatric disorders. So there's a great lab that was presented through the ANS Education Committee, an online thing that's run at UNSW, where they use crickets and they put them in a little plastic container with a clear lid that's got a grid on it, and they kind of they score grid crosses as a surrogate for movement, and then they actually shoot up the crickets with either a stimulant or a depressant, and do the same thing again and see the actions on the nervous system. And so the person who lectures that, Lauren Harms, is really keen to implement that lab. Again, that's sort of autonomous to LTP, to the LT, but we'd like to at least put it on the platform, even if we're not actually making measurements. It'd be great if we could get a camera over it and we could do movement tracking. And Melinda and I have talked about that. I think that's fairly sophisticated, but I think it'd be an awesome opportunity for us in neuroscience if we could do that. And then finally, in week 12, as decision making and reward, and there's an LT experiment, classical conditioning. This would be difficult to run in the lab because you need a participant who doesn't know the paradigm, and then you give them a scare. And so if every group is doing that, then all of a sudden the secret's out. So we're going to have to do that as a demonstration with one participant up the front. And that's what the lecturer is proposing to do there. But then that lecturer also does a lot of work in animal models with rodents in operant chambers. And so she's going to get some video footage of how you might do a similar sort of experiment in an animal model, and probably bring some of that video content into LT and embellish the second half of that classical conditioning. That's all the slides we have. I'm sorry if we did go over time. We wanted to get as much out there as possible because we want feedback if anyone has any thoughts. So back to you, Melinda. Thank you. Thank you.
Yeah, thanks very much.
Thank you very much, both Brett and Alan. That was yeah, really good to hear your yeah, plans. I actually have to say that I was, like, in all honesty, I was very skeptical about the EOG, and just seeing it, I was so convinced. You know, it just out of the box, you see the signal, and it just so convincing. I was so skeptical. I was almost like a third-year neuroscience student, just you know, thinking that it's not going to work. But then you see it's just so clear. I was really impressed. I think that's a really powerful one. Yeah.
Yeah, it's a beautiful sine wave, right?
Yeah.
Because that's what you sort of the stimulus when you're just rocking the person from side to side. Well, not rocking them, but turning them from side to side. Yeah.
Yeah, and the difference between what you expect to see and because you expect to see your eyes to do something, but then you see the trace and it's doing something else.
Yeah.
Yeah.
No, you're right. The extended stimulus. Yeah, it's sort of a nice way of getting to understand a very important sensory system like balance, of course, that unbiased, but it perfectly demonstrates the kind of physical limitations that it has to work under. So
yeah. I did have a question. Does any of the panel have a question?
I have a question about whether the crickets work instead of the cockroaches, because the cockroaches can be hard to get, and I wondered if crickets were a more reliable source or bigger legs, or I mean,
I've seen there is, I mean, in the teaching literature, I mean, typing into PubMed neuroscience undergraduate teaching, I have seen run sheets for using crickets for that lab, probably not in LT, maybe not even in LT. And I think there might be someone again in either UNSW or USyd that uses crickets. So I think it's certainly a reliable source. Look, I'll have to get back to you. The day that Melinda was up here, I bought a box of crickets, but they sat in the corner of the lab and chirped as we did all the other things, and we didn't get to them. So you'll be happy to hear I
anesthetize them. With the cockroaches, we just put them in the freezer, and that anesthetizes them.
Yeah,
is it the same
and that's what we would be proposing to do. I think you do that. You take that same approach.
Okay. No, that would be good. I
guess I can get back to you on that. Definitely, Brian.
If they come around, they're going to jump, which is a bit of
that's right.
Cockroaches, which when they come around, they just move.
But they are smaller, Brian. There's no question about that. So if you're dealing with smaller cockroaches, you'd find these perhaps a little more challenging than small cockroaches.
I got the large.
In Europe, we used to have locust. Does anyone do? I'm not Australian. Does anybody have locusts in a shop or something? But locusts were one of the other invertebrate preps that they used to use in physiology labs.
Right. Okay. It's good to know.
Years when we've been in a bind, we've used I can't remember what they're called, but we just call them the rainforest beetles. It's a simple principle.
Okay.
Found just as long as you find a beetle of some kind with a big enough leg, it seems to be pretty robust.
Okay. Because like Brett mentioned, we can't get ethics to do mammals in the lab at all. So, you know, we've done earthworms and we've done leeches and snails and all sorts of things. But and when I was in the UK, we also used to eat chilies in the lab. That was
good.
That we did, but that was more of an ion channel receptor type thing rather than temperature sensation. But we did do it and then have a bit of yogurt on hand to show it that it blocks the receptor. But so we did that as well. So it's interesting I'll be interested to see what you end up with for that. It's great.
Many thanks.
I just had a question about the second year EEG prac you did. Did you have any trouble with the placement as Kate asked earlier? Like, because I imagine the second year students are a bit more easily discouraged.
It's
then
a bit, look, it's a bit fiddly, but again, the course coordinator for that course, Phil Jobling, I'm in the lab every week, but Phil kind of runs it. He really has this ethos where the best thing is where it's difficult and students need to troubleshoot, and really that overlay is something that's really rammed home. And in fact, even though this is second year, we don't give a lot of instruction, so we don't have a detailed run through on what they'll need to do. The weeks I do it, I can't help myself, so I explain it, but Phil loves to just let them go and see how well they navigate it. And you know, they're second year students who don't like reading instructions, and then you wander around. I was trying to, there's some classics where there's one group that went through the whole lab and they hadn't resized the axes so they could see the data. So they'd done this elaborate experiment that took nearly half an hour, and I walked over and they didn't even know. I said to them, guys, you must just think you're so lucky and good. And fortunately, they hadn't even seen if the transducers were working. But that's a great learning experience.
Yeah, that's learning.
You've always got the backup of, as Brian said, the example data that you can give them if you need to. You hope that those sort of profound moments will really shape their futures.
Cool. This is all really good discussion, but I think we should move on to Kate. If that's okay, and we can always carry on.
I'll stop the share. Sorry, that's the important thing I've got to do.
Oh, good, yeah.
I'll stop the share. Oh, it's over here. You are stop share. There we go. Sorry.
Oh, by the way, I wanted to mention I do have that video of doing the EOG. So if at the end we could look at it if anyone's interested, with your consent, of course.
Oh yeah. No, I've forgotten that you took it, Melinda.
Yeah. I have it in the back. If anyone wants to see it, I can pull it up. Cool. So next up, we have Kate Thompson from the Molecular and Biochemical Therapeutics at the University of Canberra. And her expertise is in visual neuroscience and ocular development. And she's been a very distinguished lecturer. So I'm really excited to hear from her. Do you want to take it away, Kate?
Yeah, thank you so much for having me. I just want to reiterate what the other guy said. I think this is a great idea. I'm really excited to sort of hear what everyone's been doing, because not many people at UC actually use the Power Lab system. We're trying to push more and more people into using it. So it's good to actually see other people using it and hear what they've been trying. So a bit of context about what we're doing while I work out how to share my screen. There we go. Has that come up?
Yep. It's black. Oh, yeah, now it's up. Yep.
Yeah, excellent. So the unit we're currently working on is called Excitable Tissue Physiology. We have no role in naming this. Essentially what it is it's the neurophysiology unit that we teach into our medical, biomedical, and general science courses at UC. We do a little bit of service teaching into the health faculty. A few of their students come across for this unit, but a lot of them have moved over to their own equivalent unit now. So this is a third year unit. However, it is the first time most students have come across any neuroscience in their degree here. So we do still have to sort of treat it as a neuroscience 101 unit, because most of them have heard of an action potential, but that's about it. So we've sort of built it around neurophysiology and the concept of, maintaining homeostasis and how the nervous system works to do that. So the first half of the semester is sort of built around general principles of the nervous system. And then the second half of the semester, we sort of move through various systems of the body and look at how the nervous system, especially also in combination with the endocrine system, work to regulate homeostasis within each of these systems. So we try and keep it relatively physiology based. A lot of our students are just going on to either medicine, public service, working in analytical labs, or onto further allied health degrees. And so we sort of are trying to, cater to all bases. And what we've got is we've got a very, what we call it, but like maybe a bimodal sort of cohort. We have those students who are really engaged, really want to do all the work, would come to spare lessons if we had them. And then we have the students who are like, this is second semester, end of my degree. I just want to get through with the minimum trouble possible. So we have in the past had a lot of trouble with non-engagement. So like Brett and Alan, we've sort of designed to try and move a lot of our core teaching into labs, because a lot of our students are either not attending lectures, and if you look at the data, very few of them are even listening to our lectures. To the point we even actually, this semester, we're actually in a course renewal, so we're just rebuilding an old version of the unit essentially. But we actually moved all of our lectures to being sort of small recorded modules rather than having an in-person lecture, because it reached the point of it was just sort of demoralizing, and then there was no point. And we can offer probably a better product if we build it to online attendance. And therefore, all of our main face-to-face teaching is now in these labs, which unfortunately we're not allowed to make compulsory. Fortunately though, this is well regarded as one of the more enjoyable lab-based units of the degree. A lot of students come out at the end and go, oh, ETP, that was one of our most enjoyable ones, partially because they really enjoy the Power Lab system and the LT system, especially the ones where they get to actually do the experiments on themselves and see these things play out in real time on their own bodies. It really helps give them that sort of context and a bit more appreciation for why they're having to spend all this time looking at it. So it's gone really quite well. So what we've done in this iteration of the course, this is our first new version of the course, so to speak. We've kept a number of our old LT lessons that we ran. So for instance, we do the cockroach sensory nerve, which you've seen, really robust, really well appreciated. This is actually the first one we lead off on. And then we've built in a few others, removed a few, etc. So in our general principle labs, we cover firstly cockroach sensory nerve, use it to discuss action potentials and all of that. So we sort of build our labs to be around it's about a 2-hour lab. We run it a couple of iterations of it each week. We spend the first hour and a half doing the prac, and then we spend the last half hour of that doing a essentially tutorial workshop session. Some of it's discussing the questions that are ran through in the lab. Some of it's discussing lecture content that we really want to make sure that they actually do listen to at least once, and try and really reinforce the lessons that way. We haven't actually built in any direct assessment using the LT tool yet. I think it's something we want to build towards, but at the moment we're just trying to go a path of least resistance at the start, especially since we had some teething issues at the start of semester and last year, getting the LT software to probably interface with our systems. A lot of that is because our IT department has moved to a much more cautious approach, let's say. So we've had to do a lot of work in the background with like getting things whitelisted with our IT department and allowing them to actually interface between this and our learning tools. So that has been a challenge. So if anyone needs to talk about that, I'm always happy to commiserate. Yeah, so what we've done now is we've essentially got 3, I guess, blocks of content throughout the course. And at the end of each block, we have an online quiz that is focused on looking at the lessons they've learned within pracs themselves. Some of it is like, oh, what did you see here? Can you explain through the reasoning of why you saw what you saw? Some of it's explain the underlying physiology that we really wanted them to learn within the prac. And then we just have a mid-semester exam, which is all short answer. And then we have a end of semester take-home assignment, which is sort of long answers looking at each of the main systems of the body that we've covered. And we want them to do discuss how this is regulated as like a bit of a short essay style question. And then in the midst of all that, they also have a research report. I'm hoping to in the future base it to look quite like some of your guys' report systems. But at the moment it's just looking at your choose a system of the body and discuss how homeostasis is regulated, how a disease will disrupt it, and then how that's restored by a treatment. So trying to keep that feedback mechanisms, homeostasis focus. So yeah, what we do is we lead off with the cockroach sensory nerve experiment, always really well received. Sort of is a really nice way of laying down the principles of action potentials. They love this experiment. We've got to the point where this semester the students loved it so much they were grabbing cockroaches with bare hands, didn't even want to use gloves. So they really enjoy getting amongst it there. So it's very much the same thing. The bigger the leg, the better an experience you're going to have. But it is actually kind of fun to just getting them to try and troubleshoot, work out what they need to do, and get to that sort of habit of sometimes science doesn't run quite the way you'd expect. Sometimes you have to learn from each experience and optimize there. So after that, we move on to the Hoffman's reflex, because we wanted to do a reflex physiology lab. So this is a little unlike the patellar tendon hammer lab that you guys have been doing, mainly because we just don't have the hammer trigger mechanism. So we just have to work with what we had in the kit. So here you have a stimulating electrode, which you place on the tibial nerve. It's fun trying to make them find the right placement for that. That ends up being quite a bit of work for them during the prac to find like the optimal electrode placement. And what that does is it electrically induces the H reflex to cause dorsiflexion? No, the other one, plantarflexion. And you're recording the EMG associated with that. I actually ended up quite enjoying this lab because it allows you to not just talk about reflex physiology and reflex arts, because we do this in the week where we're discussing sort of sensory and motor physiology and do a lot of reflex discussions in that. You also get to reinforce the lessons that you learned in the first lab during the cockroach nerve, because they sort of are now essentially applying a similar principle, but on themselves. So you get to sort of build on that experience. And then you also get to reinforce a lot of those lessons. So for instance, you'll see the M wave, and then you'll see the H reflex, and then you get to discuss, oh, the M wave means this is just a stimulation of the motor neuron. This would not happen in a normal situation. The H reflex is the actual proper reflex of. And then you also can discuss the fact that normally your neurons are going to fire in a unidirectional manner, but this is going to induce multi-directional firing on both the new neurons. You can talk about how that's going to influence the response you get when you use higher or lower amperages in your stimulator. So it's actually quite a nice lab in terms of helping you discuss the content, and it is a nice build onto your sort of basic physiology. The main things we had with this, because this is the first time we've run this, is obviously, a lot of difficulty with the stimulating electrode for quite a few students. Some of them, it worked fine. They put it on immediately, got the response. Others had to fiddle around for quite a while, but that was a good example of, oh, everyone's bodies are slightly different. You have to experience it's a little variable when you look at biology. The other thing that we had, because we had to get them to do this sort of lying on the floor, we had them placed on just really thin yoga mats, because unfortunately we couldn't have them like hanging over tables, because this is also a microbiology lab, so like we don't really want them lying on tables. So while we had them on these yoga mats, we discovered we were getting quite a bit of electrical feedback in from the floor. We have a robotics lab underneath us, and we seem to be getting a fair bit of machine feedback. And so what we had to do is just go into the Power Lab setup file and just apply a bandpass filter to sort of filter out the feedback we were getting from that. But then after that, it worked quite well. And I think that's one of the things that probably all of us have really enjoyed about the LT software, is how much editing you can do yourself, is that you can build in parts of the lab that you really need to add, and you can remove things that maybe aren't necessary or that you don't have time for. So we've had a few things where we just didn't have the equipment and we removed that part of the lab. Or I'll talking later, we actually had one where we just had a lab that worked really well previously that we wanted to build into this platform, and we just made a whole lab using the LT tool. I'm going to go back to this. So what we then have after that is we have just talking about, we choose a sense that we want to cover. We pick vision because half of the team is vision scientists, and we have a large optometry department now at the uni. So we also borrow a lot of tools from them. So that is, we use it for the most part offline, and that is we do a dissection of a cow's eye and then run a number of visual tests. So we do visual acuity, contrast sensitivity, IOP, and then we also do a visually evoked potential demo. I think the VEPs is something that would be really nice to add in more of throughout the semester. I think it's like the students really enjoyed actually that whole experience, to the point where I ended up just sitting for another half hour to an hour after each lab with all the students who still wanted to do more VEP stuff, or the ones we didn't manage to get to because they were so excited to do it. So it's really well received, and yeah, I think you have to play around a bit with the electrode placement. Part of that is just let him fight the battle and learn those lessons. But if anyone does have anything just to make it a bit more robust, that would also probably be appreciated. But yeah, that is one of those things that's sort of limited by how many light meter pods you have. And so at the moment, since we only have one, we have to keep it as a demo system. I'm wondering if there's a potential way to do a, probably not complete one, but a relatively crude one, just by putting in the sampling rate to the machine of when a tree would fire, so they can do a sort of pseudo VEP. The latencies would probably be obviously a bit off, but just to sort of give them the general principles before going into more intense ones with the actual light meter pod. If something like that's available, that would be probably quite helpful. We also, during that lab, we do a fair bit of, just on the lab chart side of things, we do a fair lot of ERGs, or electroretinograms, in our basic research. And so we do a bit of showing them through, I guess, the trace of that and what each waveform means, so they can sort of see a bit of, oh, we're having to use these power labs all through the semester. Here's an example of them being, currently used in research. It's a nice sort of way to show them they are actually using equipment that is used in the real world, so to speak. I guess it's still at the uni, so maybe not quite. What we then have is although LT has its own autonomic nervous system lesson, we have one that we built in ourselves, just because it was really well sort of received by the students in previous iterations of the unit. And so what this essentially is we just built it in using all the same tools as before, and managed to actually just pinch a couple of the setup slides from previous, and then built in our own actual test that we wanted to do. So for instance, we start with a modified tilt table test. It's just moving from a light stand and back and forth, and getting them to try that, showing the responses they're meant to have. I have a bit of orthostatic intolerance, so then I do a demo of, this is what happens when it's maybe not working perfectly. What we then get them to do is a stress test where they set a baseline, then stand in a ice bath for a few minutes. So we have, through the power lab, we're sampling breathing rate using the respiratory belt. We have pulse rate using the finger pulse transducer, and we're also at the same time manually doing blood pressure and getting them to report all that in. And so it's a really nice way of showing those three measures working in synchronizing and working together. And then finally, we finish it off with just a simple pupillary test. We also test that during the stress tests and show that big dilation when they jump in the ice bath. And then we also just do a pupillary light reflex to finish it off as another demonstration of autonomic functioning. So yeah, I think this was a really nice experience building this in, because it really showed us just how versatile the LT platform is, that we can sort of build in things we've done previously, and have that nice continuity for students, because then they're used to this format, they know how to work it. It also saves a lot of time having them being able to input it through the LT software and have it automatically graph and stuff, so they can watch it happening in real time, which, since we are quite limited in the amount of time in our practicals, just wearing the labs so many face-to-face hours during the semester, it helps us sort of maximize getting them to do the experiment, quickly understand the results, and then still leave time for that discussion. So that's been really helpful. So then after this, we sort of move on to the endocrine system. We do a brief offline lab, just doing a general glucose tolerance test using the little finger prick and basic testing chips. I think there's a version for it in the LT software. It's much the same. We've just kept it off for now, because we want to do a have a class data set that they show plotting as we go. And we've had a few issues with our IT department letting us do a Google Sheet, because they're like, oh, then you're storing class data and stuff like that on a site that they might not have access to. And so we've had some issues with our IT on that. So we've just gone to a base, input it into a Excel sheet that's projected on the screen during the class. So then we move on to our general bodily systems. So we do the cardiovascular system, the respiratory system, and fluid and electrolyte balance and gastrointestinal system. As you guys were saying, yeah, doing fluid and electrolyte balance and gastrointestinal is quite hard these days because no one really wants to do those tests on themselves. I have had a number of other units at the uni, especially in the health faculty, have talked about doing glucose tolerance testing as their, they use it as an example of gastrointestinal just to discuss absorption. So that's, I guess, another way you can look at it. What we actually did this semester, which I'm gonna, I think next year, try and integrate into the LT system, is we did a fluid and electrolyte balance sort of mock experiment where we made a giant vat of fake urine, and then one was a sort of pseudo time course after like drinking a lot of water, drinking a lot of a hypoosmotic solution, hyperosmotic solution, isoosmotic solution, get them to do specific gravity and look at how that changes over time. And then we also made up a number of stations where we did sort of examples of alterations in urine output, like if there's blood in it, if there's excess urea, etc., etc. And then use that to sort of discuss, oh, what would this mean for what's going on in the kidneys, what might be going on at a pathophysiology level that could be causing this, and used it to sort of discuss through that. So the fake urine actually worked really well. We got much more engagement in that class than we otherwise expected, because yeah, previously that's often our least, back when we did it as a, you have to give your own sample, that was our easily least well attended class, because unfortunately students can be a bit squeamish, fair enough. But yeah, adapting to the fake urine actually worked really nicely. So then that sort of really left us with cardiovascular and respiratory to go through. So for cardiovascular, we just use the effects of exercise lab. So you have the general setup of an ECG and finger pulse transducer. And then we get them to set a baseline, look through ECG, look at how the ECG best correlates to your finger pulse. And then you do a systemic exercise. We just get them to go for a run around the building, measure the hormone after that, and then we get them to do it after hand exercises to compare sort of the what is called systemic versus local exercise. Then for airflow, we just do the airflow lab, which is just mainly the spirometry lab is what we really want them to focus on here. We also have a sort of example of chemical control of breathing that we sort of then built into this lab, where we just get them to look at breathing rate and depth when they hyperventilate, both increasing the rate and increasing the volume. And then also when they do the elevated carbon dioxide levels by breathing into a paper bag. They always really enjoyed that one. So we decided to try and add it into this lab this time. And then we get them to do the spirometry test, looking at breathing normally, and then doing an example of obstructed airway setup where they do the tape over and then breathe through. This was quite enjoyable as a number of our students have asthma, so we tried to make them not do that. We had to do a lot of warnings, I can imagine all of you do at the start of some of these labs. But that was a really good, enjoyable lab for them because they got to go and look at this. It's really difficult for me to breathe. So we saw a lot of back and forth between those students and the others in their groups. And they really enjoyed that sort of like real-time demonstration of what they experience. So yeah, it's a really well-received lab. We also have a lot of students who really enjoy the sort of tutorial part that we built in at the end. But others of them, and then for the most part, they then use a lot of the questions in the lab they use that as further what's it called? Revision. There we go. Yeah, further revision when they're coming up to exam all those quizzes. So I think that's me for now. Let's finish sharing.
Excellent. Thanks, Kate. That's really cool. Any questions from the panel or anyone in the audience?
Okay, maybe I can ask one. Just practicalities of implementing these things and safety and ethics. Launching into this and haven't yet used the nerve stimulator. Is there anything you need to do, because that can be uncomfortable, stimulating a nerve and changing the amplitude? Is there a back overlay to that? How is that negotiated for you for the Hoff reflex when they do that?
Yeah, so for us, we essentially have, I think it's like we have a list of criteria where it's like, okay, if you have any of these things, you're not allowed to do this lab, and we have to get them to sign a waiver to that effect beforehand. And then for the most part, we always do a bit of a pre-discussion at the start of the lab, going through all the general safety and everything. At that point, we need to warn you about the following things. And then we also do at the end of the cockroach lab in the week, because that's just the week immediately beforehand. We do sort of a discussion of, okay, next week you're going to be doing this. You need to, wear shorts, not long pants. If you don't want to be experimenting on yourself, you don't have to come, or you can get your other group members to do it. So it is very much a voluntary basis. And then every time that they have to do the experiments on themselves, we just have to get them to sort of sign a waiver or permission form to do that. I think if we were going to do anything more invasive, we would need to do more, but at the moment on the risk assessment and ethics balance we have, as long as it's voluntary and it doesn't actually affect their assessment, they're happy for us to do it.
And likewise, the glucose tolerance test. I like the idea of doing that. That's neat. But again, a prick and blood.
Yeah.
Is that complicated, or is that sort of covered by your current safety overlay?
Yeah, so at the moment it's covered by our current processes. We do for that one have to have an extra person in the room. So we go from, our rooms are about 30 people, so it's about we have two demonstrators in the room at a time. So we go from a 1:15 ratio to a 1:10 ratio. And then we essentially just have each person sitting in a bank, because the biggest risk we have in there is we have occasional people who faint when they see blood or have a needle. That's the main risk we have associated with that. And then the rest of it is like, you're only allowed to test yourself. You can't be testing your group mate. So we have stricter rules around it, but unless if it was going to be more invasive for I think we probably have to do more, or we might not be able to do it, but at the moment they're happy with that.
Thank you.
Excellent. I'd also want to mention, if anyone in the participants want to put their hand up and want to just ask a question, I can allow, unmute anyone as well. And while there's more questions coming, I'd like to launch a poll, just asking whether what you liked. So this really helps us see what people liked. So if you could respond to this poll, I would really appreciate it. And we'll just wait for some more questions. And then we're over time, so we kind of mix the Q&A and the session. Yeah. Cool. Kate, I also had a question, like the group data. You mentioned something about you didn't want to go to a different site for collecting group data. Is that right?
Yeah, basically our IT department had issues with us putting sort of student data on a shared website. So our best chance currently is to probably build it into Canvas, which is our main online learning tool. We just didn't have time to give that a go in our current system.
Because this is something we heard from a lot of people. So I mean, I'm just, yeah, it's good to get your feedback, because then we can, push the developers to make this a feature. Because Brian mentioned this as well, that it would be good to have like a way to collect group data. Yeah, and now also, if anyone, what Brett said, wants to, modify lessons or anything, just please get in touch with me. I'd be happy to help. Yeah, or like modify the settings files, for example. So we're actually working really close together to come up with some new, like push the boundaries of the existing pracs. So hopefully we can share some of that as well.
I can attest that Melinda's been really helpful. I mean, it's obvious, but yeah, getting him on site was really, really tremendous for us. And we'll probably try and do that again at some stage.
Yeah, that was a lot of fun. Yeah, it was, yeah.
Just a comment, actually, Melinda, jumped into my head, and this is something I've thought about a couple of times. Because we have Med-Eng students come into the integrative physiology course, I wonder, and I haven't found it, but content related to the transducers and how they're actually measuring these parameters is also another really interesting opportunity to embed some information into the system and potentially bring that medical engineering cohort into the LT system some more and into these pracs. Whether or not you sort of bifurcate, and like we have a mix, right? So we have biomed sci students sitting potentially in the same group with med-ed students doing the exercise, and they both really like it. But I think certainly for the med-engers, a little bit more content and pushing the boundaries there about, okay, so how are you measured? How is the finger pulse transducer working? How is the respiratory belt actually working? Those sorts of things might be really cool. And certainly even for the biomedical sciences students who then need to move into a domain where you are trying to innovate to measure new things and to develop new approaches, might be an opportunity.
Yeah, that's a really good idea. Yeah.
Yes, Brett,
Yeah.
we do. Sorry to jump in here.
Yeah.
But yes, we do have those sort of a different collection. So if you see our modules, there might be same lesson repeating in the different modules, but then the angle, the perspective will be totally different, though they might be doing the same experiment.
Yeah.
So for example, the case studies that we have the same case studies for the med student, and also for the same nursing students, but they have a different perspective to doing the case studies. So we do have a different, it's a user group meeting. This one was more sort of focused on very niche, which was initiated by you guys. So thanks to you. But no, that's definitely a good feedback that we can have a two user group coming together to discuss how it can be more fruitful as well.
If I may jump in, it should be easy as well, because we have data cards for all the transducers, which come with theory of operation. So you just need to get the text from there and then update the lesson in LT with the same text. So the information's there, yeah.
Fabulous.
But that's a really good idea.
Thank you.
We've got a question from the audience. This would be for Brett and Alan. So the question is, I wonder about recording EOG during Barony chair spinning. How did you keep the electrode cords untangled?
Have you got the video? Is this the cue for you to show the video?
Oh, that's a great idea. So this is actually the EOG, but maybe we'll show, I can't remember.
Yeah, I'm not sure, but that's clearly someone with a background in vestibular testing, because not many.
So this, yeah. So can
so
everyone see the video?
That was the simple test.
Yeah.
That was handy. Yeah, so that's all we have to do is just rock from side to side, or turn from side. It's not a gross movement. Now, the actual question is, all right, so now if you start spinning the person, see those leads that Brett is attached to, they'll naturally get sort of coiled up or twisted. Obviously, we can't do 10 or 15 rotations, but even by the third rotation, if it's sort of nice and relatively gentle and smooth, you're already reducing the VOR response to virtually nothing. And then obviously the dramatic event is when you stop the person and the eyes keep flickering as a result, all with the eyes shut, of course, but the eyes themselves are flickering. So you can see, thanks Melinda, that's perfect. You can see we actually took the pod off the chair, and there was enough sort of play that you can actually probably spin the person or have the person rotate 4 or 5 times, which would sort of definitely get you there. But you probably need someone above the head of the person, just making sure that nothing's happening too much to the wires. Yes, obviously they're going to get a little bit shorter, but if you bring the equipment pretty close, those wires are long enough that it prevents the wrapping. It's crude. Yes, I agree. It'll be lovely to have slip rings and all those sort of things, but that's way beyond what we can do. But thanks for the question, because it means that someone else is thinking about balance.
And I think that's how we did it, right? Someone was just holding the pod and supporting the wires, and you could actually pre-coil it in the opposite direction to some degree to get all the torsion set up to actually facilitate a number of rotations, and it really didn't confound the data at all. So you'd sort of think jiggling the wires around might sort of introduce some interference or noise, but
yeah, as long as they're sort of nicely placed on the forehead, that was great. And the other thing was that, again, these motions don't have to be heroic. They're just sort of a nice rotation slowly, which gives us plenty of time to play with the wires as they're sort of, wrapping around each other. And then obviously you do it in the opposite direction to unwind them to demonstrate what happens in the other direction. But there's lots of, I think, wonderful opportunities to ask the students, what do you think is going to happen here? And invariably they get it the wrong answer, but it doesn't matter. It's an ideal learning opportunity.
Awesome. I think any other questions that we could address? I'm hoping that this is an ongoing conversation, so that we'll continue to develop and create content and push neuroscience education. For improving it. So I guess for final remarks, if anyone has any questions or needs some help, please get in touch with me. I'm the customer success manager, so I'll be happy to help with anything you need. And the other team is our sales manager, so Parimal, or our support, Deepika, who just jumped in earlier, you met. So please don't hesitate to contact us. And I'd like to thank the panel for their great insights. It's really great to hear how you're running this neuroscience education. So I'm really grateful for joining and sharing your insights. And thank you for the participants for joining. And have a great day. And I'll close the session.
Melinda, before you go, would there be any use for a beta testing site? What I'm thinking of precisely was the changes in the EOG lab that we could potentially post up in the cloud, and people can have a look, sort of see what we've done. If they happen to want to use it, then obviously we'd be happy for them to share it. But I was just thinking, is there some sort of opportunity to share as we develop these things with other groups?
Can I take that, Melinda?
Yeah, jump in, please.
So we have done that in past. So the communities where they want to share the content for others to test and stuff like that, we can definitely host a separate playground instance for that. And then voluntarily you can put your all the content or the labs that you have created, and the others can basically review it or basically use it or whatever. So we are more than happy if there is a I think we should have that as a question as well, Melinda, here. If there is an initiative, would you like to be invited to that playground from whoever has joined, and we can share them those content of the playground. So yeah, we'll make it, and then we'll share it with whoever wants to use it. So that way we are also maintaining the privacy of a university and not letting others in.
Yeah, sure. Understood. Thank you.
Thank you.
Cool. That's great. That would be a great outcome. I guess any other questions, or happy to close the session?
No, wonderful. Thank you, Melinda.
Cool.
It and getting it all together.
Awesome.
Thanks for organizing, Melinda. It's really nice to know that there's a group of interested folks and perhaps a growing number of interested groups.
Yeah. Perfect. Awesome. Nice to see you guys, everyone.
Okay. Bye.
Cool.
Cool. Bye.
Have a good day.
Bye.
Bye.